Under normal conditions, several mechanisms, including both the innate and adaptive immune systems, contribute to homeostatic regulation in the intestine that confine the inflammatory responses to the microbiota that reside within the lumen. First, the intestinal epithelial cells that separate the lumen from the underlying lamina propria are bound together by tight junctions, here represented as red dashes, that create a largely, though not entirely, impermeable cellular barrier. Second, goblet cells interspersed among the epithelium secrete mucus that forms into a layer on the luminal side of the cells. This film-like barrier covers the epithelium and separates the cells from the majority of luminal contents. Third, at normal homeostasis, Paneth cells are thought to produce and secrete antimicrobial peptides into the luminal side. Together, these three mechanisms limit large-scale transcytosis of luminal bacteria across the epithelium. However, even at normal homeostasis, the barriers between the sides are not absolute, and cells of the immune system react to luminal contents from within the lamina propria. Dendritic cells extend processes between the epithelial cells and into the lumen, where they contact and phagocytose luminal contents and present the antigens to CD4 T cells. The dendritic cells then produce cytokines, such as TGF-beta and IL-10, which induce T cells to acquire a regulatory phenotype. Such induced Treg cells secrete TGF-beta, which in turn influences activated B cells to switch to the production of IgA antibodies. IgA antibodies produced in the intestine are efficiently transported into the lumen, where they inhibit the translocation of intestinal bacteria. Additionally, at normal homeostasis, dendritic cells can process the luminal material and become activated. Activated dendritic cells produce cytokines, such as IL-23, that can induce other cells to secrete cytokines such as IL-22, and in turn stimulate Paneth cells to release more antimicrobial peptides, which contribute to the inhibition of microbial transcytosis and minimize inflammation within the lamina propria. This forms a homeostatic regulatory loop that maintains the inflammatory response directed toward the luminal side. Focusing on the details in Paneth cells, it is thought that cytoplasmic sensors such as NOD2 can detect when material derived from microbiota enters the epithelium. Muramyl dipeptides derived from bacteria activate NOD2, which in turn delivers signals to the nucleus and induces the production of antimicrobial peptides. These antimicrobial peptides are then released from Paneth cells into the gut lumen. Thus, many active processes on both sides of the epithelium confine microbiota to the intestinal lumen, limit intestinal inflammation, and thereby contribute to normal homeostasis. However, in certain diseases, normal homeostatic mechanisms break down and lead to an exacerbation of the inflammatory response to gut microbiota. For example, polymorphisms in the human NOD2 gene are associated with Crohn's disease. It is thought that mutations in NOD2 decrease its ability to detect the presence of material derived from the microbiota. Without NOD2 activation, the NF-kappa B pathway remains inactive, and antimicrobial peptides are not induced. This weakens an important mechanism that helps maintain homeostasis under normal conditions. When minor infiltration of the epithelium occurs, the mutation in NOD2 prevents the induction of antimicrobial peptides, which may allow local transcytosis of microbiota into the lamina propria. This invasion of the lamina propria requires a more intense immune response, which involves the activation of macrophages, dendritic cells, and T cells. The stronger stimulation of dendritic cells and macrophages leads to the production of cytokines such as IL-6 and IL-12. Antigen presentation by dendritic cells to T cells causes the differentiation of naive T cells into the pro-inflammatory Th1 and Th17 subsets. The inflammatory activity of these subsets contributes to the development of the granulomatous inflammation characteristic of Crohn's disease.